Okay, before we begin, are there any questions? Can somebody confirm you can hear me?
Yes, we can hear you.
Thank you. Oh, didn't get it in time. There we go. All right, let me share my screen. Not very useful. All right, today is February 27th, almost the end of February. And we are doing Lab 9 today, identification of gram-negative organisms. This will be our last real lab. All of the future labs will be working on the unknown project, and your unknown project is due Saturday. I can't read that. March 9th. The unknown notes pages will close on Friday, March 8th. If you do request a test on Friday, make sure it's a test that will incubate for only one day, because if you ask for a test that incubates any longer, you will not get the results in time. Any questions about any of this? All right, let me go ahead and close that down. And let's talk about Lab 9. Lab 9 is the identification of gram-negative bacilli. You do have some reading, but not very much, about a page on biochemical tests and bacterial identification. And then 3 or 4 pages on classifying and identifying microorganisms. There's a video clip in today's lab. Actually, there's several, but upon the learning objectives today, upon completing this lab, you should be able to, 1, interpret the results for the following biochemical tests for the IMVIC reactions. And IMVIC is an acronym for 4 different tests. I for indole, M for methyl red, V for Voges Proscure, and I may be butchering that pronunciation. I'm not Russian. I don't speak Russian. And C for citrate test. We'll also talk about the carbohydrate fermentation tests, and those are a number. What they are all the same, but they're fermenting a single sugar. And instead of calling it glucose fermentation, lactose, sucrose, whatever, I'm just calling it carbohydrate fermentation. We'll also talk about the motility test and the hydrogen sulfide production test. To be able to explain how to interpret TSI test, that's the triple sugar iron test, the SIMS deep test. SIM is sulfur indole motility test, and then the growth on McConkey's agar and HE or hectone enteric agar. McConkey's is MAC abbreviation. All of these tests are tests that run more than one test at the same time. So you'll get multiple results. Triple sugar iron runs at least 3 tests, maybe 4. SIM runs 3 tests. McConkey's and HE, well, McConkey's runs 2 tests, and HE, I think, runs 3 tests. I'll have to talk about that. We'll talk about those. And then use biochemical analysis to solve case studies involving gram-negative bacterial infections. You'll have a number of case studies to solve in today's lab. Any questions about what we're doing? All right, members of the family Enterobacteriaceae are small gram-negative bacilli, and they're non-spore forming. They are all facultative anaerobes. They all ferment the sugar glucose. They're all oxidase negative organisms. This is one of the largest families of bacteria. This family has 53 genera. 24 of these genera have been known to cause human infections. And here we're listing some of the normal, common genera, which are part of the normal microbiota of most, well, a lot of people, not all people, obviously, like Salmonella isn't part of our normal microbiota, but it is part of the microbiota of animals, particularly birds. And these, we call them, they're usually the microbiota of the intestinal tract, and we call these organisms coliforms. They are opportunistic pathogens in that they can cause other infections, including urinary tract, wound infections, pneumonia, and septicemia. So if they grow in regions outside of the intestinal tract, they may cause an infection, and we call them opportunistic, meaning they are may be part of our normal microbiota, and growing elsewhere, they cause an infection while they're opportunistic. Common opportunistic infections in human include members of Escherichia, that genus, especially Escherichia coli, E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera are frankly human pathogens, and they're associated with gastroenteritis. These include organisms in the genera Salmonella, Shigella, and then certain strains of E. coli like Escherichia coli, Suravar 0157H7. These antigens, 0157H7, are human, what's the word I'm looking for? Not pathogens, but molecules that can make us sick. We're going to just word it that way. There's a term for that. I've forgotten it at the moment. But 0157H7 is a specific strain of E. coli, which causes gastroenteritis. It differs from the normal strains of E. coli, which are in our gut and don't cause us any problem. And in fact, they actually help us digest our food. Any questions about any of that? All right. Similar to the family Enterobacteriaceae, there's the family Pseudomonaceae. Pseudomonaceae aeruginosa is a human pathogen. However, we generally call it an opportunistic pathogen because it does not tend to infect individuals who are of normal health and have a normal normal intact skin and mucous membranes. So it's called an opportunistic pathogen because it can cause infections, especially in immunosuppressed individuals, burn patients, and trauma patients. Well, immunosuppressed individuals don't have much of an immune system, which is why Pseudomonas aeruginosa can infect them and then grow in them. They don't have much of an immune system to resist it. And then the burn patients and trauma patients frequently have their skin or their mucosal membranes damaged. And so it can get in the patient and then infect them. Like I said, a normal individual, their intact skin and intact mucous membranes tends to prevent Pseudomonas aeruginosa from getting established. We have talked about Pseudomonas aeruginosa before. It is implicated in healthcare acquired infections, which we've labeled before nosocomial infective agents. And this is one of the nosocomial infective agents. It's not the main one, E. coli is, but it is a major one. And it particularly hits burn patients and trauma patients. Pseudomonas aeruginosa tends to be in any environment that's wet, including wet soaps, diluted detergents, or disinfectants. I've always wanted to test the disinfectants in the lab. If you were to go into the real microbiology lab, we have disinfectants on the table. And I've always wanted to test them to see if they have Pseudomonas aeruginosa growing in them because, well, once it gets established, it's hard to remove, and that disinfectant is in the lab all the time. Anyways, family Pseudomonas is similar to the family Enterobacteriaceae. They're all small gram-negative bacilli. They differ in that they are all non-glucose fermenting organisms, and they're all oxidase positive. Any questions about any of that? All right. It's important for microbiologists to be able to identify and perform tests on gram-negative bacilli, especially when they're given to a clinical lab to be a clinical specimen to see if there's something causing the disease in the patient. And here's a whole bunch of tests that are frequently ran on gram-negative bacilli. And here's a device. And what you do is you have a pen and you take this metal pen and you get bacteria in the tip of the pen, and then you push the pen inside right here, and then you inoculate 12 tests as the pen goes all the way through these different tests. We won't talk about most of these tests, but we'll talk about a few of them. Any questions about any of that? All right, the first test we're going to talk about is the indole test. Some organisms produce an enzyme, tryptophanase, that converts the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added to the tube, and Kovacs reagent reacts with indole to produce a red color at the surface. E. coli tests positive for indole. Klebsiella pneumonia does not test positive for indole. So tryptophan, in the presence of the enzyme tryptophanase, makes indole, which when you add Kovacs reagent to it, it's supposed to be a cherry red color. And this photograph, I think, is off color. That looks more purple to me. It should be closer to a cherry red color. Any questions about the indole test? Positive and negative result, no change in color, and the indole will be, the Kovacs reagent will remain yellow. All right, let's move on to the methyl red and Voges Prescure test. These two tests are in the same tube. And what's frequently done is an individual inoculates one tube of MRVP test. They then incubate the tube. And then after incubation, they split the tube into two portions. One portion is used for the MR test. The other portion is used for the VP test. Any questions about any of that? Both of these tests are looking at the end products formed from glycolysis. If acid products are made, we can determine that from the MR test. And if a neutral product is made, we can determine that from the VP test. All right, MRVP contains peptones and glucose, and it's usually inoculated for 2 to 5 days. Let me see, the MR test requires 5 days. The VP test requires 2 days. And usually what we do is we just grow it for 5 days, and then it's good for the MR VP test. You could run the MR test at 2 days, but if it's negative, you have to repeat the test and then run it for 5 days, because some organisms just do not show up at 2 days. You got to incubate it for 5 days. And in my procedure, like the unknown project, if you request the MR test, I'm just going to always let it incubate for 5 days. All right, so the MR test, you just divide the tube into two. One portion you use for the MR test. All you do is you add methyl red to the broth. Methyl red is a dye and a pH indicator. If the pH indicator remains red, then acid products are made, and so we say the test is positive. Let me go and show you a result. There's the MR test after you added methyl red, and it's red, so that's MR positive. There's a methyl red test after you've added MR, and you'll notice the MR turn yellow. And usually that color change happens immediately. And so that's MR negative. Any questions about the MR test? All right. The other portion of the tube is the VP test, or the Voges-Perskure test. You take that portion of the tube, you add Barrett's A to it, shake it, you add Barrett's B to it, shake it. You're shaking it to bring in oxygen. Oxygen is needed by this test. And then you let it sit for 30 to 60 minutes, looking for a color change of red, usually on the top of the tube. If the tube turns red, it is VP positive. And like I said, you have to let it run for 60 minutes maybe before you can say it's negative. If the tube does not turn red, it is VP negative. What you're looking at is these reagents react with acetone, a neutral product made from glycolysis. And if acetone is present, the reagents will turn red for VP positive. Any question about that? All right. Let's move on to the citrate test. Oh, wait a minute. Before we move on, let me say one thing nice about all of these tests is the indole test, the MR test, and the VP test. All three of them turn red if they're positive, and they're all yellow or no color change if they're negative. Any questions about that? All right. Let's move on to the citrate utilization test. This test is used to determine whether the bacteria tested can use citrate as its sole carbon source. So in this tube, the only carbon is the molecule citrate, which, if you remember, is in the Krebs cycle shown here. And if the bacteria can utilize citrate, you'll see growth on the tube, which I don't see there, and then the tube will turn blue. If, on the other hand, the bacteria cannot use citrate, it will not grow on the tube, and it will not change color. It will remain green. What happens is when the bacteria are growing on the tube utilizing citrate, they make a base as an end product, and the base will switch the pH indicator from green color to blue color. Any questions about any of that? All right, E. coli and Shigella are citrate negative, and Citrobacter fruendi is an organism which is citrate positive. I don't know if I have any others there. Nope. All right, any questions about the citrate test? Let's move on then to the PR carbohydrate metabolism test. This test is a tube that has one sugar in the broth. And if the bacteria can ferment that sugar, it will make acid products in fermentation. And the color will change from a reddish or pinkish color to a yellow color. This we call acid, so we abbreviate that A. That would be negative or K. K stands for alkaline. Negative just means the test doesn't work, isn't positive. If it ferments, it does produce acids because that's what happens when sugars ferment with the bacteria. But sometimes it may ferment and change acid as well as ferment and produce a gas. And if you see a bubble in this inverted Durham tube, the tube is filled with media. If there's a bubble in there, that means gas was produced from the fermentation reaction, and that one is abbreviated AG for acid and gas. It's called the carbohydrate fermentation test because we're fermenting a carbohydrate, but as I stated, we only have one sugar in the tube, and it could be glucose. You just have to check and see what the sugar is. Could be glucose, it could be sucrose, it could be mannitol, could be lactose, it could be inulin, it could be another sugar. I think those are all the ones that we could test, but it could be another sugar. You just put one sugar in there, and we call it the carbohydrate metabolism test because all the tests are the same. The only difference is the sugar that is metabolized, and we'll call it fermented. So instead of calling it glucose metabolism test or the sucrose metabolism test, I'm just calling it the PR carbohydrate metabolism test. Any questions about any of that? All right. One problem with this test is it also has protein in the media. And once all the sugar is fermented, especially if you have a very quick growing organism like E. coli, if you incubate it for too long, all of the sugar will be metabolized. And then the bacteria, instead of being inactive, it'll start utilizing the proteins in the media. If you metabolize proteins, you make the amino group is pulled off the protein, and then the amino group is excreted as ammonia. Ammonia is basic, and that could change the color of the tube from yellow back to red or pink. And how you prevent that is you don't over incubate, especially for fast growing organisms. You should really not incubate it longer than a day. If you do incubate it for 2 days and you put in a whole bunch of growth, you may metabolize all the glucose, and then you start metabolizing the protein, and then that can revert the color. So it was yellow, but with more time, it starts becoming pink. This is not a problem in our unknown test because I won't allow the test to run that long. All right, any questions about carbohydrate fermentation test? If not, let's move on to the motility test. The motility test is when we grow the bacteria in a solid media, but this is actually a semi-solid media, meaning it's very thin on the agar and it sort of wiggles like jello. It's so thin on the agar that the bacteria can swim through the agar. Any questions about that? Let me blow out the paint here. So when you inoculate the tube, as shown here, if you only see bacteria growing in the stab, then you know that the bacteria is non-motile. It lacks a flagella, and so it can only grow in the stab mark. If on the other hand, the bacteria flares from the stab mark, so there's the stab, and the bacteria flares out from the stab, growing something like this. It can grow differently on this side. So I'll make that a big one there. This is slightly motile bacteria. It can swim, and that's why it's flaring from the stab mark. Any questions about that? If it's very motile bacteria, it'll move further than the flares, and you'll see that the whole tube is cloudy. That's because the bacteria are everywhere in the tube, and this is a very motile bacteria. So you don't even see the stab because it's so cloudy, and the bacteria is growing everywhere. Any questions about the motility test? All right, if bacteria are motile, remember they only have a flagella. Let's talk about the hydrogen sulfide test. This test looks for the production of hydrogen sulfide gas. If hydrogen sulfide gas is produced, a black precipitate will form, and so you'll see darkening or blackness in the tube or plate, however you're growing the media. The hydrogen sulfide test may be run on different tests. You can run this checking for hydrogen sulfide production by running the triple sugar iron test or the TSI test, or the sulfide indole motility test, or the SEM test, or you can test it, hydrogen sulfide production, on the HE plates or the hectone enteric plates. On each of these, the media will turn black if hydrogen sulfide is produced. So that looks like that's a Sims test, turning black, showing the presence of hydrogen sulfide. Here's turning black on the triple sugar iron test. And then down here, turning black on the HE plates. These colonies are producing hydrogen sulfide gas while they're black. These are not producing hydrogen sulfide, and they're not black. Any questions about any of that? All right, Citrobacter, Salmonella, and Proteus produce hydrogen sulfide. I believe E. coli does not. It would be hydrogen sulfide negative. All right, let's talk about the triple sugar iron test. The triple sugar iron runs at least 3 tests at one time. These tubes have 0.1% glucose, 1% lactose, 1% sucrose in the tube. So we're looking at fermentation, and it could be of one of these sugars, 2 of the sugars, or all of the sugars. We are also looking at the production of acid. So we have a pH indicator, and it'll change color if acid is produced. We're looking at the production of gas. And then we're looking at hydrogen sulfide being produced on TSI. So we're running at least 4 different tests at once. And I'm going to count fermentation as one test because oftentimes we can't tell which sugar is fermented. Sometimes you can, but sometimes you can't. Any question about any of that? All right. For the TSI test, you inoculate it with a needle. That's shown here. And you stab, you get the bacteria on the tip of the needle, and then you stab, inoculate the bacteria into the butt of the TSI plate. You should take that down a little further, about 2/3 of the way down. I mean, it hasn't gone all the way down. You then pull the needle out the way it came in, so you have a nice stab in the TSI tube. And in the butt here, we're looking at anaerobic growth. So the growth that happens here will be anaerobic. It might not be totally, completely anaerobic, but it's at least partially anaerobic. You then pull out the needle, and on the tip, you get bacteria on the tip of the needle again, and then you inoculate the slant in an S-type motion of the TSI. And here we're growing the bacteria aerobically. And obviously, if the bacteria is growing aerobically and uses oxygen, then the butt would be very anaerobic. And then you compare the growth on the slant to the growth in the butt. So in tube 1 here, there's no change in the color, and we say K over K, meaning no change in the slant. Slant's always first, and then the results of the butt is in second. So K over K. You can also write that negative over negative, meaning no change in both the slant and in the butt. Here we have K in the slant, K, or you could write that negative, no change. And then it's got A in the butt, and that's because whenever you have hydrogen sulfide being produced, you have fermentation occurring. So fermentation has to happen before hydrogen sulfide gas can be produced. And because the whole butt is black, we know that fermentation occurred, even though we can't see it, can't see the yellow color, but you can assume it's there. If you had looked earlier, before the black totally took over the bottom of the tube, you probably would have seen yellow on the edges of the tube. Anyway, so that's K over A or negative over A, meaning it's yellow, so acid has been produced. That means at least one of the sugars has been fermented. We'll talk a little bit more about what's going on there and there. And let me say it's black, so hydrogen sulfide is produced. There is no bubble. There's no breaking of the agar. And so we're saying there's no gas being produced. Now I talked about gas, so let's scroll over to number 4. And number 4 is yellow on the slant. So that's A on the slant. Acid is being produced. The sugars are being fermented. It's A in the butt because it's yellow in the butt, meaning the sugars are being produced. Now we know glucose is a sugar that's being produced, fermented, because glucose is the base sugar. If any other sugar is fermented, glucose has to be fermented. So we know glucose is being fermented. And then lactose and or sucrose, at least one, maybe both, are fermented. We can't know if both are or only one of the sugars is, but because it's yellow in both places, we know that both glucose and lactose and sucrose are, both glucose and either lactose and or sucrose are fermented. Any questions about that? How we know that glucose and at least one other sugar is fermented is because the slant remains yellow. If only glucose was fermented, glucose is at low concentration and the bacteria grows and ferments the sugar very rapidly on the slant. So if only glucose is fermented, the glucose would be all used up on the slant, and then the bacteria will start metabolizing the protein. The protein produces ammonia when it's metabolized. That will revert the color of the slant to pinkish, but in the butt, it would remain yellow because the fermentation is happening slower in the butt, as well as the bacteria is growing slower in the butt. And as you notice, that's the way tube 3 is. So if it's red on the slant and yellow on the butt, only glucose is being fermented. If it's yellow in both, that means either lactose or sucrose or both of them are fermented. Any question about any of that? In tube 4, we also have cracking of media right there. You can blow that up to look at that better. Cracking of the media right there, and a small bubble right there, and then a huge bubble right there. So we have gas being produced in tube 4. So gas produced, no hydrogen sulfide being produced, and glucose plus lactose and or sucrose being fermented, and it's A, acid in the slant and acid in the butt. For tube 3, we only have yellow in the butt. The slant is red, so we know that glucose is the only sugar that's being fermented. The glucose was all fermented in the slant, and if we had looked at it earlier, it would have been yellow. But then the bacteria starts metabolizing the protein, making ammonia. That's a base, and that will revert the color from yellow back to reddish or pinkish. And so this tube is abbreviated K, or negative over A. Only glucose is fermented, no gas is being produced, no hydrogen sulfide is being produced. Any questions about any of that? This test is fairly difficult to realize what's going on because so many different things can happen in this one test. All right, let's move on to the sulfide indole motility test. And this is abbreviated SIM test. SIM test has 3 tests running at the same time. Sulfide production, hydrogen sulfide gas being produced, indole being produced, and then motility test is being run at the same time. So you stab inoculate the tube, and right there, we can see the stab mark in the tube. And there it is there too, and there. Right there, it's there, but you see, right there, we can see the growth is only in this tube and probably that tube and definitely that tube. That tube I'm having a hard time seeing. That's why I'm saying probably. There's only growth in the stab. So these bacteria are non-motile. This bacteria is flaring outside of the stab. All of this region is got growth, and so this bacteria is motile. This is a non-motile. This one, you can't see the bacteria because the media is black, but because the blackness is everywhere, you can assume the bacteria has gone everywhere. And if we had looked at it earlier, the blackness would have been, well, maybe probably more towards the center, and you could have seen that it had been cloudy on the edge. So this bacteria is motile. Any questions about that? So the motility test is being run. We're also looking at the production of indole, and after you incubate the tubes, you add Kovacs reagent to the top of the tube. There it is there. And you see how the Kovacs reagent has turned pinkish or reddish on these 3 tubes. That's because indole was here to react with the Kovacs reagent. So these three tubes, tube 1, tube 2, tube 3, and tube 4, are indole positive. The control tube is indole negative. And then tube 5 is also indole negative. Any question about the indole tube? All right, the last test is pretty obvious, and that is the hydrogen sulfide production test. And if hydrogen sulfide is produced by the bacteria, the tube will turn black. And there we have hydrogen sulfide being produced in tube 1. All the other tubes are negative for hydrogen sulfide production. Any question about the SIM test? All right. Let's talk briefly about McConkey's and MAC test and the hectone-enteric auger plates, or the HE plates. These plates are both selective and differential. They are selective in the sense that only gram negatives can grow on them. These plates select against gram positives. So if you see growth on either plate, you can assume that the growth is gram negative. Any question about that? They are also differential on their ability to ferment the sugar lactose. If the bacteria can ferment the sugar lactose on McConkey's, the bacteria will take on a purpley, pinkish purple color, and the media around the bacteria will also be purple pink color. If the bacteria cannot ferment the sugar lactose, the bacteria will be their normal color, and here the bacteria is either white or off-white, and there is no coloring of the media. So that's the McConkey's media. Remember, gram-positive will not grow on McConkey's. If the gram-positive, if the bacteria cannot grow on the McConkey's, you cannot assay for the lactose fermentation. On hectone-enteric agar, we already mentioned you can test for hydrogen sulfide production, and if hydrogen sulfide is being produced, the bacteria will be black in color. If the bacteria cannot produce hydrogen sulfide, then they will be their normal color if they do not ferment the sugar lactose, which in this case will be white or off-white. If the bacteria can ferment the sugar lactose and it's growing on HE plates, then the bacteria will be salmon colored. Salmon is a yellow-orange color, meaning the lactose has been fermented. If the bacteria cannot ferment the sugar lactose, there will be their normal color, which here is once again white or off-white. If you have both hydrogen sulfide being produced and the sugar lactose being produced, the colonies will appear to be black. So you cannot tell from the colony, but as you can see, the media around the colonies, let me blow this way up. The media around the colonies will turn salmon colored. Like right there, the media around these colonies are salmon colored. Actually, it's almost a pink orange color, but and the media, you go by that to determine the lactose being fermented if the colonies are black, because you can't see the salmon color in the colony if it produces hydrogen sulfide gas. Any questions about that? Oh, too far. Go back.
Dr. Volpe,
Mhm.
I think I'm unsure of how to interpret this because it's two separate tests, but are they always run together and therefore your result is always the two of the tests together?
Are you talking about McConkey's and HE, or are you talking only about HE?
McConkey's and HE.
No, these two tests are not always ran together. Okay.
Okay.
Oftentimes you'll only be running McConkey's, other times you'll only be running HE. Would you ever want to run both of them together? Maybe, like you might want to run HE to look at the hydrogen sulfide being produced. And then you run the McConkey's more to look at the fermentation of the sugar lactose. And then
Okay.
you don't even need to look for the color orange on this plate because you got it on this plate. Not
Okay.
orange,
So
but the sugar is being fermented or not fermented from McConkey's.
so plate three is the result of just the HE test? Because it
Yeah,
looks like both.
plate two and plate three are both HE auger plates. Sorry,
Okay.
that's not labeled here. It's kind of obvious from the color of the media that these are HE plates,
Hmm.
and that's a non-lactose fermenting bacteria that does not produce um, hydrogen sulfide. This is a non-lactose fermenting bacteria that does produce hydrogen sulfide. This is a bacteria that does not produce hydrogen sulfide, and that's not labeled here, but it is a lactose fermenting, and that's what the LF stands for, which is right there.
Okay.
Yeah.
Thank you.
All right. All right. So you're not going to be physically performing any of these biochemical tests because everything we're doing is going to be online, but you will be held responsible for knowing how to perform and interpret each of these tests. We have video clips on the carbohydrate fermentation test here. We have a video clip on the SIMS test here. We have a video clip on the MR and the VP test here. And we have a video clip on the triple sugar iron or TSI test here. And then we have a video clip on the McConkey's auger test there. So watch each of these videos going through the different tests. And then when you're finished with that, click on this. I don't know why that's so small. Click on the microBuzz site to open up that web page. And you only need to look at the differential tests over here. And you only need to look at the tests to help you fill in the table in the worksheet. So the first question on the worksheet is to fill in this table. I've done the very first cell for you in the oxidase test. Let me blow that up so I can read it better. A positive result of oxidase positive, the cells or the spot where you put the reagent turns blue. The negative would be obviously not that. Okay. And then fill in the test for a positive and negative result for all of these different tests, the indole, the methyl red, the VP, the citrate. Some of these tests, you can get the information from this lab. Other tests, you will have to get the information from either a previous lab or the microBuzz. Oops, I got the wrong browser here. The microBuzz website. Like the citrate test you actually get from this lab, but like the urease test, you can open that up and get the results of the urease test from the microBuzz website. Boy, that's really slow. Negative, positive for the urease test. Any question about that? So you only need to look up on the microBuzz the tests that you need the information for filling in this table on the worksheet. Any question about question number 1? You do need to fill in both the positive and the negative result.
I do have a question, Dr. Volpe.
Uh-huh.
So under the selective and differential agar for the MAC and the He, are we to list the results for each MAC and He for the lactose fermentation?
Well, the negative result, you can just say no color change. You should do it for both, but if you only have it for one, I won't mark it wrong. So
Okay.
you could do just the MAC or the HE.
Okay, thank you.
Okay. All right, then move on to question number 2, and there's a whole bunch of questions to it. 2A, what reagent must be added to detect the presence of indole? So write that reagent there. 2B, methyl red is a pH indicator that remains what color under acidic conditions? 2C, the what test, so name the test, uses Barrett's A and Barrett's B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism. 2D, Simmons citrate uses the pH indicator, give the name of it, and turns blue color under what conditions, acidic or basic. So you need to add the name of the pH indicator used in the Simmons citrate test right there. And then you need to tell me what the blue color is under either acidic or basic conditions. Now, to give me the acidic or the basic, you could write it here, that's fine. You could also bold acidic or underline acidic or bold basic and underline basic. So whatever your answer is, you must make it clear. And do note that on 2D, you do have to answer 2 parts to this question. What the pH indicator is, and then at what conditions the indicator turns blue, either acidic conditions or basic conditions. Any questions about 2D? 2E is similar. You need to name the pH indicator used to detect the PR carbohydrate fermentation test. And this indicator turns yellow under which conditions, acidic or basic conditions. Once again, you can write acidic or basic for your answer over here. Sorry, that doesn't extend further. Or you could bold one or the other, or underline one or the other. But you do have 2 questions to answer on 2E. If you only answer one, you'll get only partial credit. 2F, organisms possess this structure. So name the structure, and they test positive for motility. So what cell structure of bacteria? Oh, that should be bacteria, by the way. I'm going to change this here. It's going to be too late for your worksheet, but in the future, I'll get that. Bacteria that possess this structure test positive for motility, because obviously eukaryote cells can have another structure that can make the cell motile. But bacteria only have one structure. So name the cell structure. I'm really surprised. I've never noticed that, and nobody's ever given the answer for a eukaryote cell, which would have been correct by the question, but I mean bacteria there. All right. 2G, 3 organisms that produce hydrogen sulfide are, so name 3 organisms that produce hydrogen sulfide gas. You can get that from the lab module, or probably from the microbuzz. I'm not certain about the microbuzz, how many are listed, but there might be 3. If an organism ferments glucose, but not lactose or sucrose, the TSI slant will be which color? And the deep, the butt, will be which color? 2I, state the 3 tests for which SIM is used. So Sims runs 3 tests. Name each of the 3 tests here. 2J, MAC and HE plates are both what? In that they inhibit the growth of either gram-negative or gram-positive bacteria. So give me what that is right there. And then you should tell me what MAC and HE inhibit the growth of either gram-positive or gram-negative. And you can bold the answer here or underline it, or you could write it out here if you want to write it. Any questions about that? And then the third part, so that's J1, J2, and J3. They are also what? In that they separate various bacteria based on visual differences from sugar fermentation. All right, any question about any of that? And then you have some case studies to do. The case studies are in the lab module. I'll show you the case studies in a minute. So read the case studies, and then this is the key you're using to solve the case studies for this lab only. Only use this key for this lab. Do not use another key. Do not use a previous lab's key. Use this key to solve the case studies in this lab. Do not use this key to solve your unknown project, because for one thing, this key only has bacteria from 2 families, Enterobacteriaceae and Pseudomonaceae, gram-negative bacilli. We'll come back to this table in a minute. And then when you read case study 1, use this table to write down the results. Like if case study 1 tells you that the oxidase was run, then put the results for the oxidase test here. If the glucose fermentation test is run, which it probably was, put the results there. If the lactose fermentation test is run, put the results there. The TSI test, put the results there, and you may need more room to explain that. Hydrogen sulfide test being run, put that there. For like TSI, you could get the hydrogen sulfide. You don't need to talk about the hydrogen sulfide here. Probably on the TSI, I'm intending for you to go put the color of the slant over the color of the butt. So you could have like negative or negative, or negative over A. So maybe that's what I'm meaning here, because you don't need to put the hydrogen sulfide there. I don't see a place for gas being produced, so we're probably not looking at gas production here, but you could discuss that there. And then the motility test, with the results for the motility test, positive or negative, indole, positive or negative, MR, positive or negative, VP, positive or negative, citrate, positive or negative. So for all the case studies, put the results in for the different tests here. On some of them, you may not run, and probably most of them, you will not run all of the tests. Just leave the cell blank if that test is not being run for this case study. And then write the species that gives you the infection for case study number 1. And once you get the results of case study 1 in the table here, then scroll up from this table looking at it to this key here. For example, if oxidase was negative, it did ferment the sugar glucose, it did ferment the sugar lactose, the TSI was A over A, the hydrogen sulfide production was positive, motility was positive, MR was positive, VP is negative, and citrate is positive, you know you have Citrobacter frundi. So write that in the worksheet for that case study. Any question about any of that? So you could write that there, for example. All right, let's take a look at the case studies. Let me show you where you can, well, actually, in this case, I have the case studies in the lab module. So let's go back to the lab module. Case study 1 here. You can read about the case right here. These are the different tests being run. It does tell you the organism isolated on BAP. You can get the hemolysis test here, as well as characteristics of the bacteria. The organism growing on MAC. So if it grows on MAC, obviously it means it's either gram-positive or gram-negative. You could look it up. And then there's a color here. So that's telling you the differential test of the McConkeys. And there's the gram stain. To see that, you got to blow it up. So let me blow that really large. That's about as large as I can get it. And there, right there, you can see one cell, there's another cell, there's another cell. They're small, but you can make out the shape, and you can also make out the gram stain. Of course, I've already mentioned all the bacteria in this lab are either in the family Pseudomonaceae or Enterobacteriaceae. So they all should be gram-negative bacilli. And they should be all pretty small bacilli, but definitely bacilli. So I told you the results of the gram stain, but pretty obvious because that's all that's on this test. Let me shrink this down a little. And then after they grow up the bacteria from the patient, they then run a bunch of tests. And sorry, this test has been split there. This is the SEMS test right here in A, the MR test in B, the VP test in C, the citrate test in D, the TSI test on E. So record the results of these different tests and then figure out which species is causing the case study. Case study 2 is here. I like this one because Mikey talks his mom into buying a pet turtle. He does well at changing the water, but he's not very diligent about washing his hands after touching the turtle. 2 weeks later, Mikey develops sickness, and they do some tests on Mikey. We grow up the bacteria on McConkey's agar, so it obviously can grow on McConkey's. The colonies of interest are shown by a arrow. So that colony of his interest, and that's a white colony, not a pinkish colony there. And I'm not sure, these must be both colonies, white and pink, because there's a full pink colony and there's a full white colony. And then they grow the colony of interest on the HE plate here. You can see the color of that, what the results mean. And then we have different tests ran on day 2, the results of the SEMS test, the MR test, the VP test, the citrate test, and the triple sugar iron test. So fill in the results of your test and then figure out which bacteria was isolated and write the species that is making Mikey sick. Where's my worksheet? Down here in case study 2. And do that for the rest of the case studies. Are there any questions? If not, I will turn the lab over to you, and I will be here until 4:30 or the last student exit the lab. And remember, I am here to help you with any questions you have in the lab. Any questions? All right, so get started. Go. All right, go ask your question.
So I finished figuring out what my unknown is For the final report, do you have like an example page of it? I feel like I remember you showing us an example of the final report, but I can't find it anywhere.
If you look in the lab 5, let me show you this. Portion of the lab, you can find an example of what the lab report should look like, and it's called, I think it's called Sample Journal. See how slow my computer is? I'm not sure if that's the internet or my computer. It's gotten very slow. So week
It
5.
could be that you have multiple tabs open too.
I'm sorry, I can't hear you.
Sorry, it could be that you have multiple tabs open too.
It could be, but that usually isn't a problem in the future, in the past. Anyways, there's the example, the sample journal. Okay,
Okay, thank you.
all right.